cannot join the meeting. She said that it show pending request or something like that.
Oh, it's still pending request. Is it? I know.
Another one. My friend is Eng.
But no, there's no request anymore inside here.
Hmm?
All the request full already, you see? But I'll just
Well,
pending.
I
Yeah,
asked her.
there's no more requests. So far, there's no more requests. Now, just a minute. I'm going to share. This is my view. So basically, there's no more pending requests.
Okay. Thank you, doctor.
Mm. All the total members we have right now in this group is 128. Okay, you can ask her to reapply or, but yeah, there's no pending request right now. Okay, so are you guys ready? We will just, I'm just going to go through the group later on. Oh yeah, your quiz. You can answer it later on. Quiz, it is going to be open until 4 o'clock. I'll try to finish within 1.5 hours. And then you guys can proceed with your quiz later on. Okay. So first group 1, group Sarawak. Okay. Oh, all full already. Now anyone from group Sarawak 1, okay, which you haven't really found any group yet. Anyone from this part A of Sarawak, like Kuching, Samaran, Syrian, Sri Aman, Sri K, Sibu? Because I noticed that it is all full. Okay, I'm supposed to create more groups. But yeah lah. But yeah lah. Over here we have Lawas, Miri, Muka, Limbang, Bintulu. These are quite full. This one full as well. Carlson Francis, are you here?
Yes, doctor,
Carlson.
I'm here. I'm here,
Carlson,
doctor.
kau mind tak if you can join group Sarawak 2E with
Huh?
Florence,
But
Gretian?
doctor, on my laptop screen, it states that group Sarawak 2A has 3 people, including me inside. So that's why I choose 2A. But I don't know how.
But
Yeah,
from
it.
my, is it? Your
Yeah.
your 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 observation is the latest kah? Pagi ni.
Yes.
Serious?
Oh, no, 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 no. Okay, okay. I
I
just refresh
think your
my
friend
tab.
left you already.
Yeah, yeah. Never mind, never mind. Right, right, right.
Okay.
I see
You
that
go to
already.
group Sarawak 2E, ah.
All
Okay,
right, sure,
then
no problem.
okay. Okay, for Sabah we have Sabah 1 full already. Full. Okay, so group Sabah all full already. Nice. Group, this is what? Selangor, Melaka, Negeri Sembilan. One. So for Eng Xiaoying, Kwek Fan Ching. Okay, are you guys here?
Yes, doctor. And about Eng, she is the one she showed me the picture that the screenshot that she's sending the request and haven't
Oh, the
accepted
pending.
yet.
Just a minute. I check again. Wait a
Oh,
minute.
thank you, doctor.
Still no request lah.
Why? That's weird.
Yeah, still no request yet at
That's
the
weird.
moment. From my side, it's like no request right now when people are, yeah, not yet. Maybe it's her internet problem, connection problem.
Maybe.
If you guys think that Microsoft Teams platform is not good enough for us to proceed with our lesson, you can just tell me so. I can change to Zoom or WebEx or any platform to your likings lah. Cuba refresh. Dah refresh dah. Satu kali dah. Still no request. It's all right. Okay, I'll just go back. So later on, if Eng came in and Kwek, you two, can you guys move to join group SMJ4 with Pham, Yenro, D, and Choi?
Okay also I think.
Okay, huh?
But
You guys are not enemy, right?
Not enemy, but I think some of them are from Selangor because I know two of them.
Oh, and you are from where?
Melaka.
Oh, from Melaka. Selangor, Melaka still yeah, a bit distance lah, but within 1.5 hour driving, right?
Not really.
Iya kah?
2 hours, I think. 2 hours,
Yeah
I
lah,
think.
1.5 or 2 hours, you speed a bit, then you reach already lah.
Okay, okay.
Yeah, I mean like, yeah, still consider that, but if you are considering like from Kuching to Lawas, it is like 1.5 day drive or around 14 hours drive. Like from one corner of Sarawak to the other corner, so it's like 14 hours. It's even crazier. That is like continuous driving for today. If we stop for resting, probably it will go whole day already. So yeah, it's like okay Selangor and Melaka not really that far lah, can lah.
Yeah.
Go in,
Okay,
join them.
thank you, doctor.
Okay. Or if you guys can discuss if you think that Selangor, Melaka quite far between each other, then you choose a site visit area in Negeri Sembilan maybe, so that both the team can actually go to that place lah. Okay. For your either discussion or not discussion, but maybe either a representative from either you or Eng, you guys can represent your group for the site visit.
Okay, thank you, doctor.
Okay. Untuk Nur Suhaiza, Christina, and Aiman, you guys are 3-member group. Maybe you need to have another siapa untuk join. Apa pula? This is Perlis and Kedah. Tiada orang dalam Perlis dan Kedah. And then we have this is Pulau Pinang and Perak. Pulau Pinang, this one full already. Okay. Terengganu, Muhammad Fidaus. Ada kat sini kah? Muhammad Fidaus bin Muhammad Fikri?
Ada doktor.
Fidaus, bolehkah kau pergi join yang tadi? Saya group mana?
Selangor tu
Ya, Selangor tu. Kau pergi join mereka boleh. Tapi Terengganu dengan Selangor, lah jauh lah. Tapi dah lah. Kelantan juga tiada orang kan? Rasa Kelantan ada, tapi Dafu juga. Ya terpaksa lah. Mungkin kau kena sacrifice sikit lah. Atau kau jadi representative pergi bahagian Terengganu, mana-mana farm dekat Terengganu ke, kau boleh collect your own data and then you share with your group pun boleh.
So masuk group mana satu tu?
Yang tadi yang dengan Selangor tu kat mana? Kat mana sudah Selangor masuk dalam group Nur Suhaiza, Christina, Aiman punya group boleh?
SMJ7
SMJ7.
Alright.
Aiman, kau orang okey kah untuk terima Fidaus? Jangan bermusuh-musuhan. Tiada jawapan pula. Okey lah rasanya. Kalau mereka tak okey tu kau beritahu saya, Fidaus.
Okay.
Okay, so Kelantan, okay, satu group full already. Okay, Pahang also no group, and then Johor we have one group. Yeah, one group over here. So yeah, okay lah. Four members group still okay, still boleh. Okay, acceptable. Okay, so remember one thing, since we select you, you guys have selected your group according to the state that you belong to, rather than your group, your own lecture group 1, lecture group 2, lecture group 3. So whenever you submit your proposal later on, your presentation later on, or later your letter your formal letter, remember to bracket kat belakang group lecture yang keberapa. Get what I mean or not? This grouping is for state grouping, like group JHR3. Tapi when you submit your proposal or your assignment, remember to bracket behind your own name. Mungkin Farah Muyanti datang dari group lecture 1. Nur Farah datang dari group lecture 2. Nur Nadia datang dari group lecture 3. So you have to state it. Okay, so that later on it's easy for me to at least trace your name lah. Kalau tidak, mampus saya just want to trace your name in order for me to key in your marks. Okay, for the sake of me. Okay, make my life easy a bit. Okay, ada lagi ke? Tak apa-apa. I was in Perlis and Kedah. I shift my choice to Kelantan. Okay. Okay. Records. Boleh lah. Okay, I think that's about it. Kalau tiada apa-apa, we just start with our class. Now, today's class will be on lecture. Lecture there. Lecture 2. Okay, now today's lecture will be about tools of animal genetic engineering. Okay, tools of animal genetic engineering is posed until you turn to share window. Okay, sharing is posed. Apa I'm not sharing. Wait a minute. Okay. Okay, now this is my slide. Okay, so lecture 2, tools of animal genetic engineering. Okay, now before we begin, just this is a very brief introduction into the genetics, the development of the genetic field. Okay, how does it actually come about? Okay, since we have our classical punya genetics done by Mendel, whereby that he did a monohybrid crosses, dihybrid crosses, and then he came out with all the dihybrid crosses, monohybrid crosses punya progeny, F1, F2 generation ratio. Okay, from there, what actually happens until today, we can have the advantage or have the techniques to even clone our gene, to modify our gene, to produce a genetic modified organism, into produce even human who have been modified, their genetic have been modified, okay, previously, although it is controversial thing to be done, but somehow scientists actually managed to do it. Okay, so eventually in what exists in sci-fi series, maybe we can only imagine people in the military try to create superhuman by using genome editing, by using gene enhancing or doing cloning. Okay, but somehow this actually occur in real life. So how are we going to deal with it? Okay, those two sisters who have been born with their genome edited, are we going to treat them as normal human being individuals, or are we going to treat them as special human beings with certain extra ability with them? Okay, we know about that two Chinese sister punya case, whereby their genetics have been reorganized, whereby they are being resistant to HIV infection. Okay, so they can, does it mean that they can have a open lifestyle? Okay, where they can have multiple partners at one time? Does it mean so? Okay, so what is the ethical consequences? What is the social consequences of it? Okay, should they be treated the same as us? Okay, are they considered as special human? So now, before we start with the lecture, we have to go through this one. Now, after classical genetics, after Mendelian's law, we have this 1903, whereby Walter Sutton actually found that gene actually resides on chromosome. Now, what he did was that he noticed that during meiosis of sea urchin, there's some organelles which actually pack themselves, okay, into this X shape. Now, this organelle, when the cell divide, before the cell actually managed to divide, this chromosome has to basically separate first. Okay, once it is being separated, once these organelles has been separated, only then the cell can separate. So they were thinking, if they are going to take out this chromosome, remove this chromosome from the cell, does it actually hinders the whole process of meiosis or cell division? Okay, and they prove it right. Okay, so they managed to confirm that gene actually resides on chromosomes. Okay, in 1903, and fast forward 1910, Thomas Hunt Morgan begin with his genetic mapping or genetic crosses experiment. What does it mean by genetic crosses or genetic mapping? Now, this Thomas Hunt Morgan is the first person who managed to use dihybrid crosses of Drosophila fruit fly. Okay, basically use this, as you can see over here, this PR represent purple color code, purple color eye, if not mistaken, purple colored eye, and then PR positive over here represent red color eye. Okay, and then we have this VG. VG stands for what? I think either the body segments or the wings. I think the wings lah. Okay, so it has a different wings. Now, after crossing, somehow this crossing, now he uses initially he uses this. These are the true breed. This is true breed. Purple vestigial punya body size. Purple. This is red. and another opposite punya trait. Okay, these are true breed, true breed. These true breed, when cross over, this is the F1 generation. Okay, now as we all know, if we breed true breed with true breed, cross it over in the F1 generation, what are we going to get? We are going to get all heterozygote, whereby only the dominant characters will be represented, correct or not? Now, over here in this F1 generation, this purple color and this vestigial wing punya individuals are all represented because the recessive traits are being covered, are being masked. Okay, you know the theory of dominance and recessive, right? Okay, now this F1 generation later on are being crossed, okay, or test cross, we call it, through an experiment called test cross. If you know your general genetics well, then you will still remember what is test cross. Okay, test cross whereby the F1 population we test a population, okay, a population of unknown individuals, whether or not they are homozygous, dominant, or heterozygote, because the phenotype only show dominant characters. So we want to test whether they are heterozygote or homozygote. What do? We do test cross. Test cross with what? What kind of genotype? The true breed of the recessive trait. Okay, the true breed of the recessive trait. So they cross them over. Now, the interesting part comes. Okay, in a normal, if according to Mendelian law, this kind of test cross, it is supposed to produce recombinant progeny. The ratio of recombinant progeny is supposed to be 1, 1, 1, 1. Okay, 1, 1, 1, 1. But somehow, this is what Thomas Hunt Morgan got. Okay, 1339, 1195, 151, 154. So to his surprise, he was like thinking, hey, this is not true. Because if, according to Mendelian law, the ratios of all these individuals through the test cross, you can find out what is a test cross later on. Through a test cross, they are supposed to be represented by ratio 1:1:1. So what actually happened? So after certain statistical analysis, he noticed that genes are sometimes not actually absolutely crossover. Some genes are actually linked. Okay, when it really depends on the distance. Okay, distance of the gene. So using this kind of method, he managed to map what is the distance. He gave a so over here, this is the calculation. This is the formula that he actually used. Recombinant frequency, recombination frequency. What is the recombination? What is a recombination frequency? These are the recombination progeny, recombinant progeny. PR plus VG, PR plus VG means to say red eye, vestigial wings, and purple eye with no vestigial wings. Okay, these are the recombinant progeny. These are the original punya the true bred punya genotype, the parental genotype, we call it. So you remember or not? Because parental genotype from here. This is red eye, no vestigial wings. So this is one parental genotype. This purple eye with vestigial wings, another parental genotype. These are considered the recombinant progeny. So Thomas Hunt Morgan observed, these two are supposed to be in the same ratio as the parental genotype. How come it is not? Okay, the phenotype is supposed to be the same, 1:1:1:1, according to Mendelian law. So after certain calculation, notice that these two genes are actually linked together. These genes are actually linked together. Okay, the crossing over are not absolute. If it is totally random, it should be 1:1. Okay, the proportion is definitely balanced. But over here, it is not balanced. Due to this unbalanced linear distribution of the recombinant progeny, you can actually map it onto a single line. Okay, and then you can predict how is the distance. So he predicted the nearer the distance between two genes, the lesser the probability of them being crossover. Get what I mean or not? Because in crossover actually happens during your meiosis, and meiosis actually allows crossing over, recombinant progeny to be produced, because sister chromatic with homologous chromosome actually cross over in order to exchange or swap their genetic material during homologous recombination. So over here, if certain genes, this purple eye and this vestigial wing punya gene, they lie too close to each other. So the probability of them crossing over, to be segregated, okay, has been reduced. Yeah, what I mean or not? So through this, actually, they can actually propose like using landmarks or milestones, can just landmarks lah, on a single line. Then they start to map a genetic map, okay, according to statistical ratio. You will be surprised, okay, you will be surprised. The work that is being done maybe 110 years ago, okay, 111 years ago, today this kind of ratio still applies, okay. People still use this kind of ratio in our marker assessment, genetic marker assessment, genetic markers mapping. Okay, we are going to talk about genetic markers next week and the marker assisted selection another week. Okay, but over here it is very important for you to understand this particular concept. Okay, now fast forward again. From 1910, Thomas Hunt Morgan has done his work. We have 1940s. Okay, starting from 1940s, there's the number of breakthroughs. Okay, Avery et al. Proved that the chromosome, within the chromosome, you know that inside the chromosome there are DNA, bound together with histonic protein, correct or not? Okay, so chromosomes are not just DNA. Inside the chromosome there's histonic protein, thereby it is being bound together. Now, then within this era, Avery actually proved that Avery and Hersey and Chase, they actually proved that DNA is the genetic material rather than the protein. Why is there such a doubt? Okay, we have to remember in DNA we have only 4 different nucleotides. We have cytosine, we have guanine, we have thymine, we have adenine. Okay, so there are only 4 nucleotides only. Okay, so no matter how you recombine it, the segregation just doesn't account for so many traits that you can observe. Okay, in human, you can not only in other complex organisms, there are just so many traits that is being observed. So just 4 different nucleotides. How does it actually account for so many kind of traits? The combination or the probability or the permutation just in doubt. Okay, it's just in doubt. Okay, so somehow they actually came out with the theory that DNA is the genetic material rather than a protein. But you have to remember, protein, the basic subunit are amino acid. At least there are at least 20 plus type of amino acids available. Okay, so if you, according to permutation, at least you can have so much of a different characters or different types of protein being produced. Okay, and then we have the discovery of role of DNA stimulate more genetic research by Delbert, Chargaff, Crick, Monod. All these, they have different findings as well. Chargaff ratio, you know what is Chargaff ratio? You have anti-parallel, you have this double-stranded DNA. Okay, whenever you do a double-stranded DNA analysis, the ratio of the cytosine always equivalent to your pyridine. Cytosine always equivalent to your guanine. Okay, and then your adenine always equivalent to your thymine. And then we have Crick. Okay, Watson and Crick, you know, they came out with the double helix model. Monod, one gene, one enzyme theory. Of course, these days we have all these alternative splicing, blah blah blah. So, but his monod contribution to the molecular biology is significant. Okay, in the 14 years between 1952 and 1960s, okay, the structure of DNA was elucidated and genetic code cracked, and the process of transcription and translation was described. Okay, and then we have this. This is a very important degeneracy code table. Okay, that you have learned during your general genetics as well. Okay, whereby we have all for each of the amino acid, they happen. Researcher at that time managed to map what kind of code. Okay, is it does one single nucleotide account for one type of amino acid, meaning to say there's 4 nucleotides. So how does this 4 nucleotide result in the different arrangement of protein with 20 plus type of amino acid? Get what I mean or not? Okay, the ratio doesn't add up. Okay, so the genetic code, is it possible that it is a dinucleotide codon? Dinucleotide codon means to say two nucleotides arrange themselves to define one single amino acid. Is it true? Okay, if that is the case, 4 power by, is it 4 power by 2? Yeah, 4 power by 2, only 16. 4 times 4, only 16. 16 types of the combination, it still doesn't add up to your 20 types of amino acid. Okay, so it has to be something different. Is it possible that it is a triplet code? Genetic code is a triplet code, so 4 power by 3. 4 times 4 times 4. Okay, so you have 4 different arrangements. So if you have your paper with you, so you can plot for a triplet codon, we have 3 sites. Okay, the first site we have we have 4 nucleotides possibly occupying the first site. The second site also 4 different nucleotides possibly occupying that second site, and the third one as well. Okay, so you know the permutation 4 times 4 times 4, it will be 64. Okay, at least 64 accounts for more than the 20 amino acids. If that is the case, then some amino acid would be sharing the same triplet code, correct or not? I don't know whether you know what I'm talking about, dabbling about or not, but yeah, you just, yeah. So we have this, you can see uracil, cytosine, adenine, guanine, and you can put it together. Okay, so UCU, uracil, cytosine, uracil, defines serine. Okay, uracil, cytosine, cytosine, serine again. UCA, UCG, serine again. Okay, this particular codon, how do they actually decipher it? They decipher it, I think you learn it in your general genetics. They use this tRNA model, okay, whereby there is different level of deciphering it. I'm not going to talk about it. Cannot remember also. Okay, so then fast forward again from 1960 plus to 1971, 1973, another new technology actually being developed. That is the recombinant DNA technology, whereby you can join DNA together. Okay, join DNA together, and then through this particular process, we actually allows gene cloning to be performed. What is gene cloning again? The process of multiplying a particular segment of DNA, okay, or the desired gene that you want in vivo. Okay, you can produce it, mass produce it. Okay, remember one thing: in order for you to study a genetic structure, the molecular structure, or a lot of this physical structure of this gene, it actually requires abundance of DNA template to work with. Okay, so gene cloning actually provided the aspect onto mass producing copies of DNA. Okay, and to allow deeper understanding and deeper work on it. Okay, and then fast forward again, 1980s. What had the another invention was by Carey Mullis on the polymerase chain reaction. Okay, polymerase chain reaction. We are going to study polymerase chain reaction in next 2 weeks maybe. Okay, so polymerase chain reaction, a very important technique as well. Until today, during this pandemic breakout, we are still using qPCR. You keep listening about our minister, the health minister, the MOSTI minister, talking about PCR, polymerase chain reaction, polymerase. Why is it so important? Why don't we just use our RTK kit for the detection of COVID? Isn't it simple and fast enough? Some people say it is not that accurate, okay, because of certain issues. Why? Why is it so? Okay, so why do we still want to rely on qPCR, real-time PCR? Okay, at the beginning of the outbreak, the pandemic last year, so everyone was fighting over this qPCR. Okay, the hospital were asking everywhere, do you guys have a qPCR, whether we can borrow or not, or we can we can use it or not. Okay, so qPCR is very important technique. We are going to discuss about it. What are the alternative? What are the different techniques that derive from polymerase chain reaction? Okay, polymerase chain reaction doesn't just add on with your tag polymerase, primers, nucleotides, and then hoo-ha, you have your DNA. Nope, there are a lot of different applications to it. Okay, so from 1980s onward, we have DNA sequencing. 1990s, beginning of 1990s, we have the first effort, okay, the kickstart on Human Genome Project. right? Human Genome Project. It took around 10 years for the Human Genome projects to be completed, to be wrapped up, but it is not a complete human genome, okay, yet, okay, at that time. But right now, it is still quite considered. There's no 100% of completely deciphering a human genome. Okay, probably you can get near 99%, 98% to it. Okay, so in the year 2000, they wrap it off. Okay, after 10 years of kickstarting the Human Genome Project. Okay, just imagine 10 years to sequence a genome. Now, these days, with DNA sequencing is going forward to next generation sequencing. These days, even third generation sequencing. You can actually sequence the whole genome within days of time, not even days. With like third generation sequencing, you can have your genome sequencing machine being placed on your desk. Okay, you can sequence it, okay, just in front of you at your office or at home. Okay, you can sequence as long as all the reagents is available. You assemble all the reagents, put it into into that particular it is like a hard disk kind of shape or a phone kind of shape, maybe slightly larger than the phone. Okay, this we call it the Oxford nanopore. Okay, nanopore. Okay, or the Minion. Okay, yeah, the name is Minion. Okay, so that sequencing machine, you can actually plot it into your desktop or your laptop, and then you put in all the DNA with all the reagents inside. It can sequence for you within a few hours time. It can complete a whole human genome, but of course the resolution would be lesser if you compare to the large big Illumina machine in the lab. Okay, now that is genome sequencing human genome. And then what else happens in 2012 onwards? 2012 onwards, we have this, which I'm going to talk about. We have these two fellows over here. Okay, these are the two researcher, Emmanuel Carpentier and Jennifer Doudna. Okay, they won the 2020 Nobel Prize Award, but their findings was back in 2012. Okay, what is their finding about? CRISPR-Cas genome editing. Okay, just now we talked about gene cloning. We use huge chunk of DNA, put it into another organism, and then we try to multiply it. Okay, or sometimes we want to express it. Now, in this particular case, we are using CRISPR-Cas genome editing. Now, rather than cloning it, putting the whole gene inside, now we can start to twitch. Okay, it is kind of like, it is analogous to you when you are being asked to complete an assignment. It is this whole rather than copy and pasting the whole text, it allows you to do small twitches within this whole chunk of text. Okay, bit by bit. Now, what kind of point? It actually allows mutation, or not mutation. It allows deletion, gene knockout. Okay, or to do single nucleotide polymorphism, single nucleotide changes. Okay, just one single nucleotide changes, because sometimes in our gene coding region there are certain single nucleotide polymorphism which changes the behavior of our protein, which later on results in a different phenotype. Okay, now these techniques actually allows the twitching of your DNA to a very single nucleotide level. Okay, you can twitch it so that the behavior can be changed. Okay, later on we are going to look at some of the examples that were being produced using these particular techniques. Okay, there are some techniques, okay, whereby they actually produces different animals using these techniques. Okay, we are going to talk about it. So that is fast forward to 2020. Okay, that is the latest findings and adoption of biotechnology. Okay, of course, in the middle we still have what we call it marker assisted selection. Marker assisted selection is more like a combination of artificial selection, like all this breeding selection that we have been practicing since like 1,000 years ago, together with molecular biology. Okay, so rather than changing any sort of DNA genetic code or any gene within an organism, we try to tag them. Okay, tag the chromosome or the genome of these individuals, and then pre-select them so that their gene or the desirable traits or phenotype can be can be what do you call it? Can be inherited to the next generation. So we allow the fetus to breed so that they can produce progeny which has similar traits as the parents, which is desirable for us, which whereby we can sell them for money, which has value for us. Okay, so yeah, that is marker assisted selection. That we are going to discuss about it later on. Okay, so why gene cloning and PCR? So over here in this lecture, we are going to talk about gene cloning and then subsequently genome editing. Okay, there is a change on your slides. Okay, there's a change on your slides. I've updated in Elite as well. So last year punya slide I have already deleted it off. You refer to the latest lah. Okay, you refer to the latest because there has to be an update on the slides. So the change is quite substantial. I think the earlier part, the slides I don't change much, but the genome editing part actually adding quite a lot. Okay, it's for your sake. Now this is gene cloning over here. Basically, a small gene, a very small part of E. coli genome. Okay, you want to clone a certain part of gene. You want to study this tryptophan A gene. Okay, so what do? You cut it using endonucleases. Use a restriction enzyme, okay, or endonucleases. Just cut it into different chunks, and all these chunks you put everything into vector. Okay, on the vector itself, from the vector itself, then you do a hybridization, meaning to say you design a very small snippets of the gene from here. Okay, just a very small snippets of it, a conserved region. Desirably, it's a conserved region lah. Okay, in particular, a conserved region whereby we design a fluorescent probe. Okay, so that later on, after you have cloned all these genes, okay, you transform it into E. coli. Okay, transform it into E. coli. So there are going to be hundreds and thousands of colony that is being formed, whereby each of these E. coli harbors different segments of genome. Then you will be asking, how do I find out which one is the gene that I desire out of these thousands and thousands of E. coli colonies? Yes, you use hybridization method. Okay, there is a Sheldon hybridization method whereby they hybridize the colony onto a blotting paper, and from the blotting paper you can use this probe, okay, to hybridize through it, and then you use X-ray or use what do you call it? Use X-ray or other radiation in order for you to detect it. Okay, so that is hybridization method. This is cloning. The cloning that actually started in 1970s. Just now I mentioned. Okay, it started in 1970s. So the process is very tedious for cloning. Nowadays, this whole cloning process maybe take around 2 days. 2 days to be done. Okay, there are kit for it. Okay, so basically we have endonucleases provided in the kit. Okay, and then we have all the restriction and then all the vector being provided as well. What is a vector? Vector is basically the vehicle. Okay, the vehicle or the nucleic acid which allows the delivery of foreign segments of this DNA into the host. Okay, whereby in this case the host will be your E. coli. So this vector helps to deliver them. Okay, once delivered in already, it allows you to pre-select out. Okay, so you create a big library. So there are thousands and thousands of colonies in hundreds and hundreds of plates, and each of these colonies you can pick up, put it into the fridge or freezer. Okay, so that you can store it for to long-term storage. Now this is gene cloning. Okay, the original gene cloning. Now these days we have also, now in 1980s we have polymerase chain reaction as well. So what is polymerase chain reaction? By using specific primer. Of course, again, you have to know certain conserved region present in this gene as well, because we want to study this gene. Okay, we don't want to clone all this. So the polymerase chain reaction basically provides an alternative or a shortcut to isolating the gene of our interest, because we want to study how does it actually behave, how does it actually undergoes transcription or translation, and then we want to observe what is the protein size that is going to be produced, what is the splicing event that is involved in this gene. We want to know about it, but without a sufficient copy number. Again, I repeat, without a sufficient copy number of this gene, we cannot proceed to any steps at all. We can't just work with one single copy. Okay, it is too minute for us to work on. Okay, so we have to multiply it, clone it. Okay, let's say or multiply it. So what do? We can use PCR. Yet the chunk, some conserved region on this gene has to be designed. We can check it through other organism. Previously, some of these genes, maybe it has been isolated, maybe it has been studied, okay, maybe partially in other organisms. We can take the conserved region within this gene, because most of the genes are homologous in nature. It has its own analog or homolog, okay, or paralog, okay. So this conserved region, we can design primers, and then we can amplify our this gene, okay, in an order of several million copies. Okay, so both of these techniques, gene cloning and PCR, why is it so important? The keyword, to multiply it. Okay, so this again, this is gene cloning again. The vector plus the fragment combined, it is a recombinant DNA molecule. This is recombinant DNA molecule. This, I'm just going to go through this very fast because you are going to do the real recombinant lab work in next semester. Yeah, like if you manage to come back, Dr. Hasnai or Dr. Rosmati, they are going to teach you on how to do recombinant DNA molecules. Okay, all this is going to be practiced by you. So at least you know the theory now. Okay, they are going to reinforce it on you, but somehow I'm just going to go through it really fast. So recombinant DNA molecule, and then you transfect or transform into the bacteria. Inside the bacteria, by utilizing or manipulating the DNA polymerase present inside, or the helicase or the topoisomerase, whatever lah, all these molecules or enzyme present in these living cells, it will help you to multiply your recombinant DNA molecules more and more. Of course, it doesn't, a bacterium or a host doesn't simply multiply a recombinant DNA molecule. This recombinant or this particular vector over here, the blue color over here, has to fulfill certain criteria. Okay, has to have certain elements in it in order for the bacterium to selectively amplify it. Bukan semua DNA masuk dia terus amplify, not really. Okay, some DNA that comes in, like bacteriophage DNA, sometimes these bacteria are going to destroy it. Okay, but what actually makes this recombinant DNA molecule so special? Although it has a foreign insert, but the bacteria, rather than destroying it, it multiplies it. Okay, which allows multiple copies of multiple copies of a gene to be amplified. Okay, inside the host. So what is a cloning vector? Cloning vector, two naturally occurring types of DNA molecules satisfy this requirement. Okay, now wait a minute. Okay, so the cloning vector, we have two naturally occurring plasmid. First of all, what is plasmid? This is a plasmid. Plasmid, you know, you learn inside your microbiology. What are the different types of plasmid? We have this replication plasmid. We have this, what do you call it? There are tons of plasmid in microbiology. I believe you have learned about it. Now, the next natural occurring types of DNA molecules are virus chromosomes. You heard about M13 molecules, M13 bacteriophage, or lambda bacteriophage. These are the bacteriophage that we can actually manipulate in order for us to put our gene of interest in, put them inside a plasmid or put them inside a virus chromosome, okay, and then let this virus do their job, or let this bacteria do their job in order for us to multiply. Again, what is the ultimate purpose of this whole event? To multiply our gene of interest, so that later on we have sufficient copy to work on, to study on. Okay, so vectors, okay, it is an agent that carry DNA fragments into a host cell, not only to store a copy of any particular fragments of DNA, but also to produce unlimited amounts of it. Okay, so the keywords over here, to carry it, the vector, to carry a DNA into a host cell, okay, so that it doesn't get to be destroyed. Okay, not only to store a copy of a particular fragment, but also to produce unlimited amount of it. Okay, so if it is used for reproducing DNA fragment, it is called cloning vector. Okay, just clone, if it is a simple cloning vector without a promoter. we call it just simple cloning vector lah. It doesn't help you to express. Now, what if this particular vector, in nowadays, most of the commercial vectors are expression vector, whereby they actually put in a promoter for you. Now, this particular promoter, when you have a gene being cloned into this expression vector, this promoter is going to help to express that gene for you inside the host. Okay, there are these different type of expression vectors available, and the host, even the hosts are being provided for you. Okay, so 4 major types of vectors. We have plasmid, viral vectors, cosmic. Now these plasmid, viral vectors, these are natural occurring. Cosmic, artificial chromosome, these are being genetic engineered. Okay, what is cosmic? What is artificial chromosome? You can find it out yourself. I think I deleted the slide already. Yeah, because I want to be fast. Because there's too much things to talk about already. Okay, I deleted the cosmic thing, like the slides. If you are interested into knowing what is cosmic, cosmic is basically taking the cos site from the viral vectors and fusing it with a plasmid, so that the cosmic actually has the behavior of both viral vectors and plasmid. Okay, what I mean or not? Okay, what is the application? What is the insert capacity? Okay, what is the size of the gene that you can actually put into a cosmic? Yeah, you can try to find it out. Okay, and then what is the host? Yeah, you can find it out. And then artificial chromosomes. Artificial chromosomes are more like they are kind of like plasmid as well, but somehow you can actually put them in these eukaryotic cells, whereby they can carry really huge chunk of DNA. For example, like 10 kb, 20 kb, 30 kb. Okay, so these artificial chromosomes. Now, these artificial chromosomes are also very useful in what sense? Whereby just now I mentioned about Human Genome Project, right? So a lot of the beginning of this bacterial artificial chromosome libraries are all established using artificial chromosomes. Okay, so artificial chromosomes are very important in the success of this Human Genome Project. You can try to learn about it. What is artificial chromosome? It has to fulfill certain criteria. Okay, so vectors capable of autonomous replication independent of the main bacteria. So the vectors has to have several characteristics. It has to have an ori site. Okay, probably this one you know already. Okay, vectors has to have an origin of replication. That is the site where your DNA polymerase is going to attach to and then starts the replication process. Okay, so from 1 molecules of DNA to become 2 molecules of DNA. So ori site is very important. And then we have it has to be easy to isolate, non-toxic to host cell. This vectors itself has to be disarmed, or it has to be rid of any toxic producing kind of gene, okay, which may kill the host instead. Okay, and then it has to have space for foreign inserts. This is very important because different vectors as now we have plasmid, the capacity of carrying an insert is different. Okay, plasmid maybe a few thousand kb, a few thousand base pair. Okay, viral vectors maybe up to 10,000, 20,000. Cosmic is around the same. Okay, artificial 10,000, 20,000, 30,000, also possible. Okay, and then unique restriction site, multiple cloning site. Multiple cloning site whereby, now what is multiple cloning site? You have in the vector itself, there has to be certain portion that you can actually cut. When you cut, you can put your fragments inside it. Okay, there's a place for you to put your insert. It can't be just a fragment. This multiple cloning site is whereby the gathering or the subset, it basically possess the subset of restriction enzyme, identify restriction site. Okay, restriction site for your restriction enzyme. Basically, you can have EcoR1, you can have BAMH1, you can have Hindi3, all these restriction enzyme cluttered in this one single multiple cloning site. Okay, this cloning site, whenever you cut, maybe you use Hindi3 or EcoR1 to cut your DNA fragment, and then you can use the same restriction enzyme to cut your vectors as well, so that later on, the hujong, the terminal end, can actually be ligated together using DNA ligase. Okay, using DNA ligase. Okay, so you have multiple cloning sites and have convenient markers. or selectable markers, or we call it antibiotic resistance gene. Most of the vectors available, we have ampicillin resistance gene, we have kanamycin resistance gene, we have what else? Streptomycin, not streptomycin, but yeah, there's plenty of antibiotic resistant gene that is available, okay, that you can actually use. Now, this is one of the thing that why people doesn't use vectors these days for genome for genetic modified organism, because in the previous days, previously, okay, so these resistant genes or these selectable markers normally are being transferred together with the gene of insert to the host. Okay, so whenever this is actually present, there is a possibility that these antibiotic resistance genes might get loose, and then it will produce certain havoc to the host. Okay, or it actually confers certain antibiotic resistant genes to the host. Okay, which later on there is a risk of maybe the development of superbug, a super bacteria which is resistant to multiple type of antibiotics. That's possible. Okay, and then vectors also has to be capable of producing multiple copies in host. Okay, yeah, it has to be producing multiple copies, either to hundreds of copies, thousands of copies, or millions of copies. Okay, this is PVR322. If you are looking at later on when you go into a lab or whatever vector that we use, most probably it comes from this PVR322. Now, what is this PVR322? PVR322 is the origin, the mother or the parents of most of the commercial vectors or plasmid that we have these days. Okay, so this PVR322 has been modified in so many ways that eventually it becomes this. Okay, eventually this now it came from R1, called E1, and all this with different combination with the ampicillin resistance gene combined with called E1, and then combined with tetracycline resistance again to become this PVR312, and eventually becomes PVR322 with a good origin of replication. This origin of replication, don't look down on it. It is very important. Why? Because it allows multiple copies of this plasmid to be produced. Okay, whether on what is the copy number of this plasmid, eventually it will go on for this. It will be accountable for this origin of replication. Okay, you can see commonly used cloning vectors. So PAC18, PAC19, okay, P blue streak, PACYC, EMBL, lambda Z, okay, and all this, most of this, as you can see here, came from this parental plasmid, okay, commercially. But it has been modified in so many ways that basically you can't really see much of the fragment left lah. So people have been working on it, and then create second version, third generation, fourth generation, until like we have all this commercial plasmid coming out. So the original sequence maybe, I don't know how many percent left. Okay, now then we have cloning vectors for eukaryotes. Most cloning experiments are carried out in E. coli as the host, and variety of cloning vectors are available for it. Okay, like plasmid, like the viral vectors that we talked about just now, this lambda bacteriophage, this M13 bacteriophage, it is being used mostly in E. coli to multiply the genes in E. coli. So E. coli is particularly popular to study the basic features such as gene structure and function. Okay, however, the aim may not be to study a gene, but to use cloning to obtain large amounts of important pharmaceutical proteins. Okay, or introduce specific characteristic. Sometimes we study gene, we don't want to just stop at the basic feature of a gene. E. coli allows us to study maybe how many exon is there, or how many introns are there, what is the splicing site available, what is the 5' UTR, what is the 3' UTR, what is the start codon, stop codon, the basic features of a gene. Okay, but sometimes, most of the time, we do not want to just stop at the basic features. We want to go further. We want to collect more of the protein itself. Okay, the gene, we can clone it into a vector, but somehow we want to express it and what it may be a pharmaceutical important protein, or it possesses certain characteristics that we want to work on. And this protein, if possible, we want to grow pure, we want to obtain a pure isolate of this protein so that later on we can grow crystal out of it. Okay, crystal out of it in order for us to run our NMR, our X-ray crystallography to determine the protein structure, this the 3D protein structure of it. Okay, we want to decipher the structure of certain proteins. Okay, so how do we do it? We have to use other types of vectors, like vectors for yeast and other fungi. Now, one of the very important yeast vector that we still use today, this what do you call it? Over here, you can have this particular. What is the name of it right here? This is this 2 micron plasmid over here is the original plasmid. Okay, it's what originating from yeast. Yeast also have their own plasmid. It's 2 micron plasmid. Talking about the size of the of this plasmid. Okay, it is a 6 kb plasmid which exists in the yeast between 70 to 200 copies. Okay, exists in yeast. Okay, so it is originally isolated from yeast. Now, how can we manipulate or bioengineer this particular plasmid in order for it to be used as an expression vector and a cloning vector and an expression vector? Okay, this is natural occurring. We want to bioengineer it so that later on it can carry a gene for us, multiply the gene. Not only multiply, it can express the gene. Okay, and then we can use a tag certain this tags on it so that we can isolate the protein for further usage, for further study, to determine its biochemical function, to determine its 3D structure, to tell us what is the overall biological function it has, and what type of motif or domains does it actually possess. Okay, so we want to know about the gene, the properties of our gene. So we can use this 2 micron plasmid to do it. Okay, so a normal yeast gene is used as selectable marker enzyme involved in amino acid biosynthesis, U2, which encodes beta isopropyl malate dehydrogenase enzyme converting pyruvic acid to leucine. Okay, which is very important, a very important selection marker. Okay, because this leucine is essential amino acids, whereby without the presence of this LEU2 gene, okay, the host itself, or we call it the yeast itself, cannot survive. It would die. Okay, so in order to use LEU2 as selectable markers, the host must be an isotropic mutant that has a non-functional LEU2, meaning to say this LEU2 is not available inside the nuclear genome of the yeast that we are going to use. Again, I repeat, the host itself, okay, has to be an isotropic mutant, meaning to say without this 2 micron plasmid, okay, without this LEU2 punya gene, this mutant cannot survive. Okay, so the plasmid has to be inside. Now, it can be tailored in such a way that it is present inside your test tube. Okay, once transformed, we culture it on a selectable plate. Okay, this type of the host that doesn't have the LEU2 punya gene, which is present in your 2 micron plasmid, this 2 micron LEU2 gene, which is not present. If it is not present, if this plasmid doesn't go into the host, okay, the host basically doesn't grow on your plate. Get what I mean or not? Okay, you can stop. If you want to ask me a question, you can just turn on your mic and ask me a question. Don't just be silent. Otherwise, I'll just blah 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 blah. Keep on going. Okay. Now let's talk about this yeast episomal plasmid. YEPs. Okay. Yeast episomal plasmid. Now altogether, there are 3 yeast plasmid that probably you want to know. There are definitely hundreds of different types, but these are the 3 basic ones that you want to probably remember. Yeast episomal plasmid, we have yeast integrative plasmid and yeast replicative plasmid as well, YRPs. Okay, so what is yeast episomal plasmid? Vectors derived from 2 micron plasmid called yeast episomal plasmid. Okay, some YEPs contains the entire 2 micron plasmid, others include the origin of replication only. So basically from here, This is the original 2 micron plasmid. They took out the origin of replication only. Okay, but most of the time they contain entire 2 micron plasmid. Okay, shuttle vector, which includes the entire PBR322 sequence and can therefore replicate and be selected for in both yeast and E. coli. Now, what is a shuttle vector over here? Basically, they combine the origin of replication from these 2 micron plasmid, because it is very important in order for you to replicate a segment of DNA in the yeast. You need an origin of replication from the yeast. Get what I mean or not? So they fuse this origin of replication of the yeast from the 2 micron plasmid with PBR322. Get it? Okay, so this PBR322 has the ability to be shuffled, okay, in between 2 different hosts, the E. coli and the yeast itself as well. Okay, so initial cloning experiment is normally done with E. coli and to select recombinant in this organism. So after selecting an E. coli, the recombinant, we can take out we can isolate the clones or isolate the recombinant plasmid or this plasmid, and then we can actually put it into a expression, the yeast as a host, since the molecule can only be expressed in yeast, or because yeast is a eukaryote, okay, which some of the genes that we study, it is eukaryotic in nature. It has to be expressed in eukaryotic cells. That's why we can't do there's limitation in using E. coli as a host. Okay, and then we have 2 other cloning vectors for use with yeast as well. We have yeast integrative plasmid, basically bacterial plasmid carrying the yeast gene, YIP, which is PBR322 with an inserted URA3 gene, okay, a selectable markers, URA3, okay, and then we have, of course, as the name indicates, this integrative plasmid, it integrates later on after the transformation itself. It basically integrates into the nuclear genome of the Saccharomyces cerevisiae, the yeast, okay, and then yeast replicative plasmid are able to multiply as independent plasmid because they carry a chromosomal DNA sequence that includes an origin of replication. Get what I mean or not? So this replication, it is totally independent, meaning to say it can multiply as independent plasmid. Okay, it carries a chunk of chromosomal DNA, okay, which with origin of replication. Okay, so let's look at the consideration and different characteristics of these vectors. Transformation frequency, a measure of number of transformers that can be obtained per microgram of plasmid. So yeast episomal plasmid has the highest transformation frequency, meaning to say whenever you want to introduce this particular plasmid into your host or into your yeast, okay, into your host, the transformation frequency will always be higher if you are going to use yeast episomal plasmids. Okay, you can transform it. So between 10,000 molecules to 100,000 molecules transform per cell. Okay, or transform cell, 10,000 to 100,000 transform cells per microgram. If you are going to use 1 microgram of this yeast episomal plasmids, is going to transform 10,000 to 100,000. Okay, now but if you are going to consider YRP, these replicative plasmids are also quite productive, giving around 10,000 transformers per microgram of your yeast replicative plasmid. Okay, but yeast integrative plasmid yield less than 1,000 transformers per microgram. So the less the transformation frequency, yeast integrative plasmid will be the lessest. Okay, the least when we're talking about. The copy number, yeast episomal and yeast replicative has the highest copy number, 20 to 50 and 5 to 10 respectively. Yeast episomal plasmid, when it is being introduced to the yeast cell, it can have during the cell division or during the DNA synthesis or during the whole replicating event, the copy number inside one single cell can goes up to 20 to 50 copies. Okay, for yeast episomal, yeast replicative 5 to 100. Okay, in contrast, yeast integrative plasmid is usually present at just 1 copy per cell, because just now I mentioned, because it is integrative plasmid, it integrates into the nuclear genome. So that's why 1 copy will be enough. Okay, so that's why yeast integrative plasmids are very stable, because over time when it is being integrated into the nuclear genome, you won't lost it anymore. Okay, as long as you to continuous select it, it is very stable. Okay, in contrast, yeast replicative and yeast episomal were extremely unstable. Okay, throughout times, if you don't protect them properly, slowly, if you don't select them, they are going to dwindle the concentration of the top copy number. Okay, eventually you are going to lose them. Then we talk about yeast artificial chromosome. What is yeast artificial chromosome? Now, before we begin, okay, chromosomes, we have to understand about the nature of chromosomes. Chromosomes, there are these three key components of a chromosome. First of all, the centromere. Okay, just imagine a single linear DNA. Our chromosomes, when it is being intertwined with a histonic protein, if you are going to unwind it from the histonic protein, it is going to be a single linear DNA. Now, what makes a single linear DNA to behave like a chromosome? You know what I mean? Making sense? Okay. What actually makes this single linear DNA to behave like a chromosome? Yeah, like eventually when we talk about chromosome, it is a linear DNA. It is not a chromosome. It is not a plasmid. It is not circular in shape. It is a linear DNA. What actually defines it as a chromosome? Okay, now three components which has to be present in order for us to define this chromosome. First of all, centromere. The centromere has to be there, which is required for chromosome to be distributed correctly to daughter cells during cell division. So remember the centromere, whereby you have your mitotic spindle, or what do you call it? It's spindle fiber, is it, to pull your your sister chromatid into different not the homologous chromosome and sister chromatid into your daughter cells. So remember it. So that would be the centromere. Okay, and then for the assembly of your mitotic spindle, and then you have your telomeres. Okay, the structure at the end of the chromosome, which also are needed in order for the ends to be replicated correctly. So basically, for a linear piece of DNA, in order for you to be defined as a chromosome, first of all, centromere. The next one will be your telomeres. Telomeres has to be very, very important. Telomeres, because it is a buffering region present at the terminal end of the chromosome. Okay, one of the very important function or biological function of this telomeres is to protect your chromosome from being nibbled away by exonucleases. Remember one thing, any linear DNA which is present inside our cell has the potential to be nibbled away. Exonucleases can actually act on them. Okay, that's why we need. Now, another functions of these telomeres is it actually function in aging. Okay, in cellular clock. Okay, whereby whenever your cell undergoes division or DNA undergo synthesis, due to the anti-parallel or semi-conservative nature of your DNA replication. So remember your semi-conservative model of your DNA replication or not. Okay, so it is very important to have these telomeres present, because if we are going to take into account what you learn in semi-conservative replication, eventually your DNA is going to get shorter and shorter and shorter. So remember, whenever we want to replicate the DNA, we have to have Okazaki fragment. This Okazaki fragment has to bind to certain place in order for a stretch of DNA to be connected or to be synthesized. Okay, but when your DNA gets nearer to determine, when the DNA replication gets nearer to the very extreme end of your chromosome, what will happen? They are going to be presented with one of the problems. There is not enough space behind at the very terminal end of this 3' end, because DNA are being synthesized from 5' to 3'. So at the 3' end, dia tidak cukup tempat sudah. Okay, am I talking about yeah. So it basically that is not really enough for space for the extension of the DNA. So what will happen? Slowly, these are going to be cut off. Okay, this is going to be shorter and shorter. Now, in nature, we have this particular mechanism to add on to this terminal end, so that at least after several cycles of the cell division or DNA synthesis, the telomeres are still protected. This batch of enzyme, we call it telomerase. Telomerase enzymes are very important aspect in molecular biology these days, whereby they study for anti-aging, anti-cancer, and other purposes as well. Okay, so telomerase is a very hot topics or hot molecular biology topics nowadays. Okay, so this telomerase help eventually to prolong this telomerase in order to protect cells from being terminated. If DNA gets to be too close to your gene already, now what will what if telomerase is not going to protect it? Telomere dah habis sudah. Okay, what will happen? This cell will undergo apoptosis. It is going to die off, or it is not going to replicate anymore. Okay, that's where you have reached the age of the limit of the age of the cell. Okay, and then later on, the third component, origin of replication, which are the position along the chromosome, which DNA replication initiate. Okay, so origin of replication, we've been talking about it. Okay, so these artificial chromosomes near origin of replication, which is also required. So these are the three elements. If you can see over here, this artificial chromosome, probably you will be asking, hey, this is artificial chromosome. How is it? Why is it in a circular shape? Okay, looks like a plasmid to me. Now, originally these are artificial chromosomes. These are commercially available. These are synthesized in the lab. Okay, so the structure of it is in circular shape. Now, in order for us to utilize it, what do? We have to, before we can use it, we have to actually cut it off using bump electron. We have to linearize it, cut off this chunk of sequence, so that we can linearize it. So the telomerase can be at the terminal end of both ends over here. My mouse is not working really. Okay, so this telomerase has to be. You guys have no question? Okay, so this telomeres has to be at the terminal end of the cell. Okay, and then we have what else? We have centromeres. Central over here. This is your centromere. Centromere has to be here. Centromere. Remember, centromere mitotic spindle has to be formed in order for sister chromatic and homologous chromosome to be separated into daughter cells later on during your cellular division. Okay, either mitosis or meiosis. Okay, the centromere is a very important aspect. And then origin of replication. This origin of replication is where your DNA polymerase is going to bind to initiate DNA synthesis later on. Okay, so once it is available, so once you have cut it off, you can put in your insert DNA. Okay, insert DNA right over here. SNA1 B1. So within this particular gene, you can cut it, put it into here. Okay, so that the gene can be carried, and then you can achieve the purpose of gene cloning. Okay, gene cloning. Cloning strategy, this one tak payah lah. Now, cloning in mammals. Cloning in mammals also got eukaryotes is not just yeast only. We have tons of it, complex organism. So mammals is we can have clones into mammals. So gene cloning in mammals carried out in 3 reasons: to achieve a gene knockout for production of recombinant protein, in gene therapy in which human cells are engineered in order to treat disease. So we can use gene cloning to treat diseases as well. That is what we call as gene therapy. Okay, gene therapy these days, I think we have multiple. Several years ago, it is quite limited, but these days I think is quite common for gene therapy. I think gene therapy even being used to I'm not sure whether it's true or not. I saw in TikTok whereby they actually use this gene therapy to treat HIV. Okay, for HIV being a patient, they can actually use this gene therapy to allow the T cells to proliferate. Okay, and then to not being destroyed by the viruses. Okay, I'm not sure how true is it lah, but yeah, all these days social media, lots of information inside. Okay, so first vector for mammals is a vector based on simian virus 40 SV40. So simian virus, normally we use viruses, okay, for mammals. Okay, the genome is 5.2 kb in size and contain 2 sets of genes, the early genes coding for blah blah blah. So the earliest that we have simian virus 40 SV40. We're going to listen to it quite a lot in in recombinant DNA technology. Okay. Now, there are different types of vectors whereby we use viruses for gene therapy, for gene cloning as well. Probably you'll be asking, why do we want to use virus? I'm not sure whether you guys have watched the Resident Evil punya movie, okay, whereby you jab yourself with this zombie becoming virus, and then initially at least can actually heal herself from what do you call these disabilities. Okay, and then somehow it turned people into zombies. Okay, so yeah, these are real. We use not like people turn into, I mean, like we use different type of viruses as vectors to deliver cells. Okay, and each of these viruses has their own characteristic. Doesn't mean that one single retrovirus we can use it for all types of all types of disease and therapy. Okay, these retroviruses, the primary host cells are actively dividing cells. If it is going to infect mammals, it is going to attack mostly actively dividing cells. Okay, entry pathway, receptor binding, membrane fusion. Okay, so vector genome forms integrated, can integrate into the human genome. Okay, transgene expression long term, it can go for several years. So the users, people have been studying it for SCID. SCID is what disease already cannot remember. Hyperlipidemia and solid tumors. Okay, so retroviruses can be used, has been genetically engineered for this kind of diseases, for this kind of gene therapy. We have lentivirus, adenovirus, adeno-associated virus, herpesvirus as well. Okay, now these are some of the, yeah, you can read it on your own. Now let's go further. Okay, CRISPR-Cas genome editing. Okay, now this is the in thing right now. Okay, CRISPR genome. Whenever you are thinking about editing a genome, okay, most probably you are going to use CRISPR, because CRISPR these days are just so convenient. You can, within 1 or 2 weeks, you can achieve your objective in modifying a genome. But somehow the whole overall process of introducing this modified nucleic acid into real cells in order for it to be proliferated, that is another story lah. But CRISPR genome editing gets so convenient these days. Okay, with a few $100, I think, or a few, maybe 1,000, at most 1,000, 2,000 ringgit, you can probably buy your own kit. Okay, buy your own kit, and if you want to do it at home, tapi yalah tak guna if you are going to, there's no delivery path. Okay, delivery path for it. But genome editing gets so convenient these days. If you want to look at the, what is this? This is, you can just Google it. It is there already. I posted the slides already, so you can watch the video on your own. How they actually, why are they being given or conferred the 2020 Nobel Prize in Chemistry? Why these two individuals? Okay, so main genome editing tools. What is the what are the three now? From genome editing to genome editing is again, as I repeat, genome editing is different from gene cloning. Okay, genome editing whereby we want to tweak it. Okay, tweak it without doing tons copying or multiplying it. We want, if possible, we want this genome editing to occur. Now, the modified genome or modified gene to be present only one single copy per cell. Okay, that is our ultimate objective, ultimate purpose. Okay, and then try to reduce non-specific targets, meaning to say, except for the intended region of the genome that we want to modify, the rest of the area we don't want it to be present in. Okay, the rest of the genome has to be totally unaffected. Okay, then it will be considered as a good genome editing. It only occurs one time, and the rest of the part of the genome are not affected at all. Now, the three mainstream genome editing tool is here. Okay, we have zinc fingers, ZFN. Okay, zinc finger DNA binding domain, zinc fingers nucleases. Okay, and then the second one is TALEN. TALEN stands for transcription activator like effectors nucleases. Okay, something like that. Okay, but the name is very long and yeah, winded. Okay, and then the third type that we have is CRISPR-Cas. Okay, Cas9 protein. This is a Cas9 protein. Okay, rather than now, what is the difference between these two? These three, ZFN and talon over here. Both of these nucleases, they uses fork1 nuclease. Fork1 nuclease. These are fork1 nuclease. Okay, CRISPR, in CRISPR punya system, we don't use fork1. Okay, it is this Cas is the nuclease that we're going to use, Cas9. Okay, so in ZFN and talon, fork1 would be the ultimate nucleases. But over here, so what is the difference between ZFN and talon? The difference lies in this zinc finger DNA binding domain. So this protein itself has the ability to bind certain motif in the DNA that you want to modify. Meaning to say, now, two main steps in genome editing is first to basically hold on or to recognize the sequence that you want to modify. Okay. Secondly, only then this fork protein, which represent the function of nucleases, can recognize the sequence and cut it. Not recognize, but cut it. Okay. So basically, you have to have your zinc finger positioning itself on the genome or on the DNA that you want to cut, and then this fork protein is going to be the scissor that cuts it. Get what I mean or not? Okay. So over here, these are the recognition site, GNN, ANN. So these are the recognition site. There is the zinc finger DNA binding domain has certain motif to be recognized, and you will be surprised. Then you probably will be asking, hey, if it has certain binding domain, so what is the frequency of occurrence of this zinc finger? Does it simply bind as long as there's a motif which it can recognize, it will bind? Not necessarily. This zinc finger DNA binding domain, the zinc finger DNA, the domain, the occurrence of it is around 1,500 only in our gene. What is it? Gene related section, gene express gene in the section, okay, in our genome. So it occurs in some of the genes only, around 1,500 of other genes that is present in our genome. Okay, so most of the genes that we have is not really most. Some of the genes that we have inside our genome has this zinc finger domain, okay, which allows this zinc finger DNA binding domain to hold on to. Okay, so the frequency is high. Okay, but of course you can try to in order, then you will say, hey, so every time when I'm going to introduce this zinc finger into my genome, so all these 1,500 is going to be affected? Not really. As long as you know what is the fragments over here which flanks the gene that you desire. Okay, the motif that flanks it, and then you can do some twitching within it. Okay, when I say motif, this motif has to have certain conserved region and certain variable region. So those variable regions are the ones that you can twitch in order to fulfill your liking. Kau mau dia target mana-mana satu gene yang ada zinc finger domain, okay, you can try to twitch this variable region. Okay, while this conserved region is going to recognize the overall that particular zinc finger binding motif. It is going to bind to it, and then later it is the fork one will cut it. Get what I mean or not? Now, similarly, this talon over here, transcription activator like factors, blah blah blah. Okay, so the main difference between it. Just a minute. Silent. Okay, I've been dragging on and on. My lunch mate is asking me for lunch already. So I think we are going to stop. Is it possible? Wait a minute. Where am I? Banyak kah? Aduhai. Banyak lagi. Aduhai. Can we continue next week? Hello guys. Are you guys still there?
Yes, no problem.
Yes.
Yes,
Okay,
ah.
okay.
Are you guys,
No problem.
do you guys understand what I'm talking about?
Haha.
Yes, Doctor.
Okay. Hey, I think why not like this? This is the basic, what do you call it, basic description of all these technologies. So you go back, you try to read on. If the rest of the slides, you can ask me for this itself, the main genome editing tools. You try to read on these three technologies. There are plenty of YouTube videos and then those YouTuber, they can talk much better than me. Whenever you listen to them with all the illustration, but probably you will understand better. Okay. My platform over here is for you. Whenever if you don't understand what they are talking about again, you can come back to me. No worries. Okay, you can take your own initiative. So yeah, no worries on it. So I'm just going to finish this part over here. Okay. So talent is as opposed to zinc finger. Okay, what is the difference between talent and zinc finger? This is the region over here. Okay, over here it uses zinc finger to identify the region that it wants to cut. Okay, to recognize the site it wants to cut. But talent uses this talent domain. Okay, talent domain over here again, it has its own residues. Okay, it is called repeat variable die residues. Okay, the name you just remember, it is called repeat variable die residues. As for zinc finger, it is this GNN-ANN. Okay, zinc finger DNA binding domain. So over here, it binds to repeat variable die residues. Okay, just remember the simple terms. No, don't have to really actually know how does it bind and all. So the nuclease part of it, once this carboxy, this tail effector DNA binding domain attached to the genome editing, the targeted region, this fork one is going to cut it. Okay, now later on. So Cas is a totally different story already. We are going to talk about it next week. You can listen to certain videos first. Now, in this Cas protein, instead of total protein that you use, this CRISPR-Cas9, it actually uses Cas protein together with a guide RNA. Okay, so it is not purely a protein. In this talent case, zinc finger ZFN, okay, they are pure protein. But over here, this Cas is the nuclease. There is a segment that for the identification or recognition of the site where to bind. It actually uses a gRNA. We call it guide RNA. What is a guide RNA? You can try to find it out. It is a recombinant DNA RNA molecules which combines a tracer RNA together with a CRISPR RNA. Okay, what are those again? No worries. We are going to talk about it next week. Now, this gRNA basically allows the detection of a region within the targeted region. Now, over here, this region here inside the guide RNA, this is the region that we can actually change. We can modify it. We can bioengineer this region, the guide RNA, so that inside in vitro, it will later on, after you have bioengineered this RNA region, it will combine with Cas9 to detect the region that you want to cut, you want to modify inside the genome. Get what I mean or not? Okay, so once it is going to be cut, this is going to undergo, wait a minute, I need to have, okay, this is going to undergo the repair system, two types of repair system. I'm not sure whether you have still remember the lecture that you have learned in general genetics, whereby we learn about homologous recombination and non-homologous end joining. Okay, now these are the two repair systems that will be used upon cutting your genome using either ZFN, talent, or CRISPR-Cas. Okay, once they cut already, by using HR, homologous recombination, or non-homologous end joining, it is going to start to repair. And during this particular repair process, then the mutation or the mistake actually comes. Okay, the mistake actually are being introduced during this whole repairing process. The cutting is not the cutting is not the main the climax of it. Okay, the real important part is the repair part, because during this repair process of the double-stranded breakage that we have inside our genome, once it is being repaired, certain changes, certain nucleotides are being lost. It is going to be gone forever. Now, if this actually occurs inside the gene, that whole gene, because of the loss of one or two nucleotides or a chunk of nucleotides, this whole gene cannot function at all. Okay, then we will achieve the objective of knockout. Yeah, what I mean or not? Meaning to say that particular gene loses its function. Okay, then we can study, or we can use, or we can improve an organism using this deletion. Okay, I'm going to talk about it later in next session. Okay, and then later on we have this SDN2. This SDN2, what is it? Site-directed nucleases. So another outcome of it is that once you have cut it already, then the genome itself, or the cell itself, will be undergoing certain other repair system to fulfill it. Okay, to fulfill the change, to put it back, what do you call it, to correct it. Okay, this correction that is being put in is error prone. Okay, or it is according to certain template which we can actually provide, and then the changes can be incorporated in. Now, this SDN2 over here involves the change of single nucleotide or several nucleotide, maybe up to 20 nucleotide. Now, SDN3, another scenario that comes out from the repair system, your homologous recombination and non-homologous end joining, is this. Okay, you can actually chop in or put in a chunk of DNA, or insert maybe a few hundred base pair of foreign insert, or 1,000 base pair of foreign insert into this region. Okay, this particular scenario or outcome, we call it SDN3. Then probably you will be asking, why do we want to complicate so much? The change is a change. Why do we still have to classify into SDN1, 2, 3? Well, this is very important. I mean, why? Because in a biosecurity and biosafety sense, okay, especially in Malaysia, biosecurity and biosafety sense, this SDN1 is very difficult to detect. A single loss of nucleotide by deletion, basically you mean like officer, or like when you try to do a very you go against the law by a little bit. Okay, polis dia biasanya dia tidak akan nampak kan what am I talking about. But over here, SDN1, it is very difficult to be detected. Okay, the loss in genetic changes, because it is kind of like natural change. Okay, without any traces being left behind. Get what I mean or not? It is very difficult for us to detect. Okay, in SDN2, again, also very difficult to detect, because it only involves one single nucleotide or several nucleotides, which always occur. This kind of polymorphism, or this kind of polymorphism always occurs inside the genome, whereby it is also very difficult. Only this SDN3 is kind of difficult, because you are adding in a whole chunk of DNA. Okay, so when we want to, not to say we want to avoid the enforcement, but SDN1, SDN2 is a hot topic right now. How do we identify a change in the nucleotide whereby it occurs so naturally that there is no trace that we can work on? Okay, unless that particular researcher, unless you guys, when you want to do it, you did slightly a deletion or a slight change on the DNA, that it becomes unnoticeable, difficult to notice. Get what I mean or not? So these are the three kind of scenario that we have. And what are the different types? Later, we are in next session, we are going to talk about how does it actually affect, how is it being detected, all these SDN, different outcomes that is being detected. Okay or not? Am I going too fast? Okay.
Okay, doctor.
Okay, doctor.
Okay, doctor.
Yeah. Okay. Now, don't you can watch back the video since it is a recording. No worries. Always you can try to ask me. And then another thing for you to be relaxed of is that this is not coming out in exam, but just that in the next week probably I'll try to postpone the quiz into the other week, so that you have the time to read and then time to ask me about it. Okay, it is not going to go into there's no since there's no final exam, but this is what I'm sharing with you is the latest technology that we have in genome editing as genome editing tools. And in order, if you are going to learn about it, you know about it, this is very critical for as a biotechnology graduates. Get what I mean? Okay. I think that's about it. So yeah, if there's no question, I'm going to share the attendance again. You take your attendance. If there's any question, you can turn on your mic and you can ask me. Any question? Yeah, you can take your attendance for those who haven't taken your attendance. Nothing to ask, or you have to have some time to digest it. If I'm going too fast. Yeah. Better. Okay. Now you all throughout the whole week, if you have any question, either you just text me, or just you just try to text me or you can, if you want to call me, you can call me, or you can gather a few friends, you can approach me so that we can have a discussion session, also possible. Okay, basically last time when there's face-to-face lecture, after the lesson your seniors are going to come to my office, knock on my doors, and then we can sit down and chit chat on what is all these technologies about, and yeah, we learn from there. But these days there's no face-to-face, so it really depends on the initiative whether or not you want to learn more about it. Okay, if nothing else, thank you so much for coming today. I'll see you guys next week.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor. Thank you, Doctor. Thank you, Doctor.
Thank you, Doctor.
Welcome.
Doctor,
Yeah.
I want to ask regarding the site visit for Sabah. We have 3 groups. So how are we going to combine since kan macam kena 2 group combine, tapi Sabah ada 3 group.
Tak apa, tak semestinya Sabah. I mean like kau boleh pilih satu, maybe a Sarawak punya group to combine with you, but maybe one would go to the poultry company in Sarawak. The other one can go to you guys probably will go into a poultry company in Sabah. Get what I mean or not?
Alright,
yes, so that's why you can that's why I talked to you guys about the different combination you can have just now. Or a belacang company as well. Since previously I think your senior also went to belacang company. So one company go to, there's different types of belacang. I think there's one red type and another type. So these 2 different belacang, they have 2 different types of processes. And then they actually 2 groups go to different belacang and then they combine and then they do a comparison. Why one is better than the other? And then what is the advantage of the other one compared to the other? So and they did a great job on it.
Okay. Thank you, Doctor.
Okay.
Doctor,
Yes,
what
yes,
is
yes.
the password for the quiz?
Oh yeah, 1755. Oh my goodness. I better post it there. Wait. 1755, 1755. My goodness, so sorry.
Thank you, Doctor.
Okay.
Good morning, Doctor.
Okay.
So that means that we can choose the other group from other states, can?
The other groups from other states?
Yeah.
Can. The partnering team. Partnering team. Yeah, partnering team from other groups can.
Okay. Thank you.
Okay.
Doctor, do we have a time limit for the quiz? Like that, within 30 minutes you must finish the quiz, something like that.
10 minutes. The time limit is 10 minutes.
Oh
my goodness. But it is only 10 questions only. Very simple, straightforward question that you can find the answer in your slides.
Okay, thank you, Doctor. And it's 10 questions, not 30 questions.
No. Crazy. 30 questions in 10 minutes. dying already. Okay?
Okay, thank you, doctor.
Okay, I'll see you. Okay. If no more question, then I'll be leaving already. I'm going for my lunch. Or you can just post it inside the chat room, yeah? I'll answer it from there. Okay, bye. Thank you so much. See you guys.
Thank you, doctor.